All right. Welcome, everyone. My name is Sam Becerra. I'm former editor of Chance Magazine. I'm glad to have Rob Cass and Emery Brown today and in a special conversation with Professor Emery Brown, who's our guest of honor. Um, and I'll say a few words about Emery and I let Rob to start the conversation. Emery Brown is the Edward Hood Taplin Professor of Medical Engineering and Computational Neuroscience at MIT, and the Warren M. Zapol Professor of Anesthesia at Harvard Medical School and Anesthesiologist at Massachusetts General Hospital. Professor Brown received his BA in Applied Mathematics from Harvard College, his MA and PhD in Statistics from Harvard University, and his MD from Harvard Medical School. Emery is an anesthesiologist statistician recognized for developing signal processing algorithms for neuroscience data analysis and for defining the neurophysiology mechanisms of general anesthesia. Brown was a member of the NIH Brain Initiative Working Group. He received an NIH Director's Pioneer Award, the National Institute of Statistical Sciences Sachs Award, the American Society for Anesthesiologists Excellence in Research Award, the 2018 Dixon Prize in Science, and the 2020 Schwartz Prize for Theoretical and Computational Neuroscience. Brown is a member of the National Academy of Medicine, the National Academy of Sciences, and the National Academy of Engineering. Professor Rob Cass is a Morris Falk Professor of Statistics and Computational Neuroscience at CMU, and an old friend and collaborator of Dr. Brown. So Rob, and Rob Cass is my PhD advisor. So I'm glad to have both of you, and especially thank you, Emery, for agreeing to be interviewed by Chance. Rob, without further ado, I'll let you to start the conversation.
Thank you. So, Emery, when I spoke at your 60th birthday celebration symposium dinner at MIT, and when I introduced you for the Dixon Prize here at Carnegie Mellon, CMU, I commented on a couple of things that have always seemed crucial to your development and success. First, your undergraduate background in applied math, which included an exposure to both dynamical systems and statistics. You saw the enormous potential for exploiting the connection between dynamical system models, represented by differential equations in continuous time, and certain time series models, state space models, that can be considered discrete analogs, where differential equations are replaced by difference equations. This has informed a good deal of your technical work throughout your career. And at the highest level, I've taken the message to be, if you want to understand dynamic neural phenomena, you should use dynamic methods, which includes both mathematical models to aid understanding and statistical models to guide data analysis. So could you tell us a little about how you now understand your own early trajectory?
Yeah, so first of all, again, thanks to Sam for setting all this up and arranging this interview, and thanks to you, Rob, for agreeing to conduct the interview. It's a real privilege and a real honor for me to do this with both of you. The applied mathematics did give me insights into dynamical systems, and at the same time I was studying statistics, and my interest in statistics was in time series analysis. So again, stochastic processes are processes that are dynamic. And I think the first time where I really saw the juxtaposition of a statistical model representing dynamics and a mathematical model representing dynamics was when I was studying circadian rhythms for my PhD. Because there was this one question that bothered me that I worked on, and I think it had me appreciate that you could have a dynamical systems representation, but then when you looked at real data, you should have a statistical representation to capture that dynamical systems sort of formulation as closely as possible. And here's what it was for circadian rhythms. You know, Richard Cronauer at Harvard had shown that a weakly nonlinear oscillator could capture the dynamics of the circadian pacemaker very well. So weakly nonlinear. So when it perturbed from its limit cycle, it comes back very, very slowly. And I was wondering, so that was a factoid which, you know, had been well established with Dick's work. But when we would analyze circadian data, we would fit harmonic regression models. So I was wondering, what was the relationship between a harmonic regression and a dynamical systems model? Because if we had that representation, then what we could do is we could fit the harmonic regression approximation of the dynamical system to the data and not just have an arbitrary harmonic regression model. And after searching around and, you know, a while, I found this old Russian mathematics book by Krylov and Bogolyubov about nonlinear dynamical systems. And they had this asymptotic expansion in there of weakly nonlinear systems. So a weakly nonlinear system has a small parameter. In this case, the small parameter multiplied the resistance of the resistance term of the nonlinear differential equation. And if you expanded that, you expanded that in a perturbation series, you got a fundamental and a third harmonic. And so that was it. I mean, that sounds like, wow, I was like, this is like so cool. So this is the model that we should be fitting. And what was interesting was when you'd fit those models, it showed you that the formulation that Dick had worked out, while it was good in a simulation sense, there was real structure there which couldn't be ignored. There was a second harmonic.
Diurnal, right?
Exactly. And the power of the third harmonic was not nearly as great as the model would predict. So it was really good because it was a direct juxtaposition of this theoretical framework, which did work very well. In fact, it was used to design experiments and make predictions. However, there were features in the data which the model weren't capturing. And I think that this was one of the things that was very critical for a lot of the experiments we subsequently did to show that light could actually alter the alter the pacemaker, could shift the time on the human biological clock. When we did those experiments, we measured where the clock was and where the clock shifted to. And to do that accurately, you really needed the second harmonic. It was, you know, you could look at it from a purely data analysis standpoint, but it was very important from a dynamical system standpoint because it actually allowed you to keep time accurately when time was shifted on the clock. So that was the first real, so that kind of prepared me.
So let me back you up though, because I'm curious if you could just say a little bit more about your undergraduate experience, because I maybe was it Fred Mosteller you got exposed to? I can't remember what you've told me in the past about that. How did you get drawn into statistics in the first place?
Well, the truth is I got drawn in statistics because of my roommates. I was, because I started off in college when at Harvard, my thing was going to be Romance languages. And I figured that I was going to be a physician that worked for like the World Health Organization or worked for an organization like Médecins Sans Frontières, because I was interested in languages and I knew I wanted to be a doctor. And my roommates were majoring in economics and they kept talking about economics and statistics. And I was like, wow, that really, really seems cool. And I decided my sophomore year to switch my major. So starting my junior year, I switched out of Romance languages and into applied mathematics. And I had to take 3 courses each of the last 4 semesters in order to meet all the requirements. And then Ken Wachter, who was at the time the senior tutor in the statistics department at Harvard, he's now at Berkeley. He said, well, you know, if you're going to go to medical school, if you want to go to medical school, you should probably write a thesis on a medical problem. And he said, why don't you go talk to Professor Mosteller? And Fred ran this very large surgical research group. This group was trying to understand how do you measure the benefits of surgery when it's something which is not done in terms of randomized controlled clinical trials. I mean, this was the sort of thing that Fred was very good at. He would take on these very messy problems, create structure in them, and sort of answer these like big questions. And the other thing was, you know, everybody wanted to work with Fred. And I was like, wow, I'm going to get to work with Fred Mosteller. I remember when I went to see him the first time, and I said, you know, Professor Mosteller, I went to see him, it was about lunchtime. And he said, you know, pardon me, I hope you don't mind if I eat my lunch while I talk to you. I said, no, but go right ahead. And he said, so I said, you know, Professor Wachter said I should come and talk to you because he felt that, you know, maybe I could do a thesis project with your surgery research group? He said, easy as pie. He said, we meet, twice a month, on Tuesday evenings over the Harvard School of Public Health. We start with a dinner, then we have a meeting after that. You're welcome to join us at the next meeting. Then there was the silence. I was like, oh, thank you. He goes, I don't think we have anything else to talk about, do we?
That's Fred, man. That's my little experience with him is so similar to that. That's amazing.
So I was like, okay, I'm out of here. I'll see you in 2 weeks, right? But that was like probably one of the most amazing experiences that you could imagine. And one was just watching Fred work as a scientist and getting people to work together, watching him think. He always led from the back of the room. He's made suggestions and he got the group to sort of think about them. And the other thing was when it wasn't like he and I had regular advising sessions like that at all. In fact, I can't remember us really formally meeting at any other time aside from that particular point. But the one thing was, I knew he was going to read my senior honors thesis. And that just made me put as much effort as possible into writing it as best as I could. And I still have the comments that I got back on it. He said it was well written. I'm thinking, Mosteller says my thesis is well written. This is huge. That's like, what more could you ask for? So just sort of seeing him do science and seeing him organize teams was a very, very formative moment, very, very formative period for me. And I am continuing to be very grateful for that experience, that advice that Professor Wachter gave me.
And do you remember the sort of decision to do the PhD part in statistics?
Well, that was the switch because I came to, again, you come to college and you think you can do everything. I thought I was going to learn all the languages of the UN. Then I shrunk it down to, the Romance languages that would cover sort of the African diaspora. And I was thinking of doing a PhD in Romance languages, And then I thought, well, statistics would be perhaps a little bit more directly related to medicine. And I realized that I should learn more mathematics since I switched into, applied mathematics in my junior year. So what I did was I wanted to learn more French because at the time my Spanish was a little more proficient. So I wanted to learn more French. So I got a Rotary Fellowship to study mathematics in France after I graduated from college so that I could sort of kill two birds with one stone, buff up my math skills while at the same time, buff up my French.
And was that remind me, was that a statistical, did you kind of study statistics at that point?
No, just mathematics.
Just math. So again,
math.
I'm just curious about the actual decision to do the PhD in statistics.
Oh, basically, as soon as I think starting my junior year, and I saw how, statistics just seemed to explain everything. I can remember during the time when I pretty much decided that my junior year after the first couple of courses I was taking. Actually, my intro statistics course wasn't in statistics. I took the introductory econometrics course for the graduate students in economics.
Okay.
And actually, that was that turned out to be a very, very influential experience for me also, because it was taught by Gary Chamberlain. And he taught it with the Groot's book, Probability and Statistics.
Oh, wow.
So he taught classical statistics and Bayesian statistics on par with each other, which was not the
norm back then.
It was not the norm at all. And I am so grateful for that, right? Because I didn't grow up with this sort of there's this thing called Bayesian statistics. It was totally on par. It seemed like something was totally feasible. And I find now that I teach my students, that kind of the same way. But it was with that junior year experience, and then also working with Fred, it was pretty clear I wanted to do a PhD in statistics. What happened was, I went to Grenoble. I spent a year there studying math, came back. And I had been accepted at Harvard Medical School, and I came back to Harvard Medical. I was accepted at Harvard Medical School before I went to France. Then I came back to join the first year class at Harvard Medical School, and the requirement at the time was then you then applied to the MD PhD program once you were already in medical school. So I did the first 2 years of medical school. I took off 4 years, did my PhD. I didn't, finally submit it until after I finished medical school. And I came back to the last 2 years of medical school. And so that's why I effectively got my PhD and my MD at the same time.
All right. So I remember, I was trying to think back to the times we first met, and I believe this was, I don't know, may or may not have been the second time, but in any case, I think we had known each other for a few years, and then we sort of bumped into each other at the 5th International Meeting on Bayesian Statistics in Valencia in 1994.
Yeah. Mm-hmm.
And I remember walking and talking with you for quite a while, and you told me excitedly about your interest in neuroscience.
Right.
So you must have started working with Matt Wilson already at, or maybe if not then, maybe shortly after that,
Right.
because, and Matt Wilson being an MIT neuroscientist, because your paper on recovering a rat's foraging path from neural recordings in its hippocampus was submitted in 1997, and it was published in 1998.
That's right. Yeah.
So that seemed like it was something of a triumph for you, in the sense that it demonstrated both the predictive power and the explanatory merit of state space modeling for understanding neural activity. So did you see it that way? And did you anticipate ultimately how influential it would be?
No, I don't know if I can honestly say I didn't think it would, I wasn't planning on it being, as influential as it turned out to be, maybe. But I really enjoyed the problem. And there, again, this was all serendipity. It turned out MIT didn't have a stat department. And I had made a conscious decision to start studying neuroscience. And because after I'd finished up, I'd been on the faculty in the anesthesia department for 2 years. And I figured I really needed, circadian rhythm is what I'd worked on for my PhD, but I wanted to branch out and have something which had a bit more depth to it. And I actually did a systematic search across the major areas that NIH was going to fund at that time. So women's health was just coming into being, genetics and genomics was starting to really blossom. There was the environment, there was cancer, and there was also the brain in neuroscience. And my friend, Steve Massaquoi, had always been telling me, Emery, you should really look at the brain. I mean, that's going to be really, really cool. And Matt Wilson had 2 papers which had come out in Science, one in 1993 and one in 1994, where he did this work showing that the place cells, these cells in the hippocampus, actually represented an animal's location in space. And that he had come up with a technique for decoding them. In other words, going from the firing patterns of the neurons to saying where the animal was in space. And so I had read that paper and I said, when I know enough, I will go and talk to Matt. He's right here in Cambridge. So at the time, I was teaching a course called Statistics in Physiology with Art Dempster at the Harvard Stat Department. And we decided to devote the second semester of the course specifically to neuroscience, not broadly physiology, but specifically neuroscience. Mm-hmm. And, so I was learning things, one lecture ahead of the students and presenting it to them. And then we got this call from Matt's graduate students, graduate student and postdoc, Ken Blum, who's now at Harvard, actually, running one of the neuroscience programs, and also Lauren Frank, who was a graduate student with Matt at the time. And they had questions about their data analysis. And so the secretary in the department said, well, Art and Emery are teaching the seminar. Why don't you guys just come over and give a presentation?
Oh, that was just coincidental?
That was just coincidental.
Oh, wow.
Totally coincidental. So Ken and Lauren came over, and Matt, finally joined in. And that's how I got started. And I said, I had planned to talk to you, but I didn't think I knew enough to talk to you yet, And so then that summer, I took the Woods Hole course, the month-long course on computational neuroscience. And I started working on the hippocampus problem, decoding problem for my project. That was the summer of 1995. And so that's how Lauren Frank and I made an agreement at that time. I said, well, if you teach me neuroscience, I'll teach you statistics, And that's how we got started. And
That's
then,
great. And you ended up on his was it his thesis committee or?
yeah, I was on his thesis committee, and then he did 2 years of a postdoc with me, before
Right.
going off to UCSF to,
Right.
take a faculty position.
Yeah, he's now a very successful neuroscientist himself. He's got a great lab. He's had a wonderful career.
Yeah, no, yeah, he's a Hughes investigator even. He's been extremely successful. But
Yeah.
I mean, it was just, serendipity. It's very, very fortunate.
Great. So then the next thing I remember was in 1998, and I think it was actually at an ENAR meeting. I'm pretty sure. I asked you to give a talk, and I asked a couple other people to give talks, and they were not very well attended because it was statisticians, and I think they didn't appreciate that this was actually super interesting. And we had lunch, and over lunch, I believe, we decided that we ought to write a review paper on statistics and neurophysiology.
Right.
And then once we got going, it turned out that all the things we thought we should be reviewing actually didn't exist. At least that's the way I remember it.
Right.
And so we kind of had to create the things ourselves, and we kind of went mostly on our own way, although we collaborated a little bit. And then it took 7 years, but we finally published this paper with Valerie Ventura that was in the Journal of Neurophysiology.
Mm.
So, and during that period, we also started the SAN meeting, Statistical Analysis of Neural Data, which turned out to be very successful. We've had a lot of those meetings subsequently. And then, of course, as Sam mentioned, you were on the NIH Brain Initiative Steering Committee. And
Right.
you made sure, I recall, and I remember talking to you during that time and reading the report afterward, you made sure that advanced methods for data analysis was one of the priorities for research, which has been, I think, really important. And then finally, most recently, you were selected as the 2020 winner of the Swartz Prize in Computational Neuroscience from the Society for Neuroscience, which is a major prize. And it's not only a recognition of you, but also it's a recognition of the importance of statistics in neuroscience. Right. So it seems like, we've come a pretty long way since that beginning point. But how do you see the current status of statistics in neuroscience?
Well, yeah, so I think you're right. I think we have come a long way, but let me just go back a bit. So the one thing which I should say when we look at the the work that I did with the decoding problem with Matt and trying to sort of go from neural spiking activity to the rat's position. So I took that problem, and by formulating as a state-based problem, where basically what you're trying to do is the state you're trying to figure out is the animal's position, and you're getting it not from continuous measurements, but from this multivariate point process, which is the spiking activity of the neurons. So basically, a one sentence summary is you had to figure out how do you build basically a Kalman filter for point processes. I think that's the simplest way to say it. And the other part about it was that, and this is where I felt that I had a strong comparative advantage with the work that I'd done as a PhD student. The big problem in data analysis in neuroscience was that people were using static methods to track dynamic processes. And so literally all I did was come along and say, well, let's just use dynamic methods here, time series techniques or state-based methods or, spectral methods that are essentially dynamic to capture these phenomena. And I think that now this is broadly appreciated, this is the way to think about it. In other words, just using static techniques and then, making some argument about why they're appropriate was not as good as, having techniques that were indeed, dynamic. And I think
because I guess you're pointing out we didn't really finish the conversation of a minute ago. Let me just add one thing, as I remember really well, I'm sure you will too. Down in Woods Hole, in fact, probably was the first time I visited there during the course that you ended up running with David Kleinfeld and Partha Mitra. Andy Schwartz was there. Yeah. And the three of us were walking and talking, and I had already, from hearing you talk about your methods with the hippocampus, I had already thought about the possibility of doing brain-computer interface with this approach. It seemed natural. And we walked and talked with Andy and sort of further developed that idea. And then I continued along those lines, and now it's like it's a standard approach to brain computer interface. That's how people do it.
Right.
It's for exactly the reasons I think that you saw initially.
Right. I think that's the case, because when and it's sort of, you have to explain why you wouldn't be doing it that way, because you have a process that you're observing, which is, the neural activity. The neural activity maintains a representation, like in the case of the motor studies you were doing, it's the movements or features of the movements. And in the problem I was looking at, it was the animal's, position, in space. And there are just constraints on that problem that make it quite amenable to being analyzed as a state space problem. And the key thing was, respecting this, the point process structure of the neural data, and in addition, respecting the dynamics. And so those constraints, pointed you in a very anyone who I think took the problem seriously, pointed you in a very, very specific direction, and sort of almost dictated what types of methods,
Yeah, and fortunately, statistics had evolved actually not super long before that to develop all these generalized linear model techniques. I mean, they were there and kind of ready to go.
Right, and in fact, David Brillinger knew this, years ago.
Mm-hmm.
He wrote about this in a paper in 1988. He basically, it's like a little one-sentence thing. Yep. Where he basically says, you can just use a logistic link function and have basically a binomial process with history dependence, and you can use that to analyze neural spike train data. It was very clear to him, because at the time he was looking at neural spike train data, and he was also looking at earthquake data. And he was quite fascinated by the idea that he could go back and forth with kind of the similar techniques, sort of for the two phenomena. And the thing that I did, you mentioned the Woods Hole course, so that was another. So there was the course in computational neuroscience, which focused on sort of mathematical modeling, and then David Kleinfeld and Partha Mitra had an amazing forethought in setting up a data analysis course, which they call neuroinformatics. And I worked with him for 10 years, teaching it and then eventually running that course, because it was much needed. I think it continues to be much needed. It closed out a few years ago, but we should go back to it. But for me, what it did was it allowed me to get a very broad sense of what was the data analysis problems in neuroscience, and also to teach some of these ideas, to a number of, young neuroscientists, graduate students and postdocs who are coming along, and sort of putting forward this idea of, using dynamic methods to analyze dynamic data, essentially. Mm-hmm. And you came and helped with that, and also, Satish Iyengar,
Yep.
from,
and Valerie Ventura.
Valerie Ventura were very loyal participants in that effort, and I think it had a tremendous impact on neuroscience, on data analysis or computational neuroscience.
And then it's interesting to think about the SAND meetings too, because I remember when the very first one, and I'm sure you remember, basically we're trying to figure out what are the problems. It was,
right.
It was the year was, I think, 2001 or 2002. And we still didn't really understand what the problems were. I mean, you had already identified one very tractable problem. Right. But there were others out there, and we were trying to figure out, trying to penetrate some of the jargon. In fact, the neural coding jargon that the neuroscientists, the theoretical neuroscientists, were so fond of, and I think you kind of started seeing through it right away. But it took a while to it seems to me, to really unpack that and to have the field kind of move away from what it was doing and towards something that was way more productive.
Well, I think the barrier to entry, and we've talked about this, several times. The barrier to entry to statisticians into this field is, it does require, if you really want to make contribution, then you really have to study the neuroscience. It's extremely difficult to be a dilettante in this field, just come in and throw a technique on something, then claim victory and then march out. It just won't happen because, one, there are a lot of very talented quantitative people in the field, and to convince them that what you're doing is a reasonable approach or the right approach, you have to argue not only from the methodology standpoint, but also from the standpoint of understanding the impact it's going to have on the neuroscience, the inferences you're going to make.
Yeah.
So you have to care about both.
Yeah.
And I think
So
that's really, really key here.
this prompts me to ask a question, because this discussion is about current state of statistics in neuroscience. Can you elaborate a little bit more about the role that training quantitative neuroscientists, statisticians getting involved with neuroscience can play in all of this? I mean, I did some neuroscience mainly because Rob was my advisor. I became fascinated by it. And, he sent me to University of Pittsburgh to sit through some neuroscience, basic neuroscience courses and so forth. But it was Rob who was interested in neuroscience, doing statistics in neuroscience methods, but not there wasn't a degree called statistics in neuroscience.
Right. Yeah, no, Rob and I have had this discussion, many times. And again, as I said, my personal bias is that there is, I think for this particular field, and it's probably true for another areas, but this one I know a good deal about from having worked in it for a number of years, is that the best way to really have impact here is to get into the field and understand the problem or the problems that you're working on deeply, from a neuroscience perspective, and then, build the methods from that perspective. Because what I found is it puts constraints on the problems that actually simplify the methodology development, for one thing. And the other thing too is, like I was saying before, being able to make a credible statement about why the method should be accepted or why the approach should be adopted, because a high fraction of the people that you're talking to, sort of don't share our perspective on statistics, and they can find very plausible arguments looking at problems from other perspectives, dynamical systems or physics or what have you, for why a certain approach should be taken. And I think if we're going to argue from a statistical standpoint, we have to know the statistics as well as the neuroscience to do that. So going back to the training, what does that mean? It means we have to train, statisticians, our postdocs, our graduate students, in the neuroscience at the same time we're training them in statistics. And that's something that, Rob at Carnegie Mellon has put a good deal of emphasis on. And I see it all the time. I see my bioengineering students, I see physics students having no fear about acquiring, the in-depth knowledge of the neuroscience. And I think we have to promulgate that culture among our statistics students as well.
So what's your, so to go back to the question that I was asking before, what is your personal opinion of, where statistics is at broadly in neuroscience? I mean, we can claim a certain number of victories, but obviously there's work to do still. What's your perspective at the moment? And do you think there's plenty of opportunity for other people to get into the field and so forth?
I think the opportunities are boundless because, one of the things that, so one, data generation in neuroscience is just, it was exploding just like data generation everywhere across a number of fields. It's not only big data, but it's big dynamic data. It's big dynamic multiple type data. So not only point processes, but continuous processes. And in addition, it's being spurred very, 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 very forcefully by the BRAIN Initiative, because the BRAIN Initiative, the idea behind it was to create the tools necessary for neuroscientists to answer questions about the brain. So which means building a lot of new techniques, so that there are a lot of new types of data that are coming out of the new recording methods. And then when you add to that something like a new phase that the BRAIN Initiative is moving to now is what's called the cell census. So the idea is to take, let's say like a mouse brain, take a region in the brain, learn the characteristics of every cell type in that region and where it projects to. So you have this very detailed anatomic information, neurophysiologic information, and now the recordings that come from that. And the idea then is to try to use that to infer the behaviors that are associated with that particular brain region or those brain regions. I mean, what I think is really cool about it is if you think of like a lot of other problems, which are big data problems, these big data problems are data which are collected in highly structured experiments. And, you make a decision about how many animals, where you're going to record from, for how long. So there's a design there. And the idea of now thinking about how you could, improve that design because, the experiment is structured is like an area which is essentially untouched. It's not being explored. So I think that the opportunities are even bigger than when we got into the field itself, because of the data explosion and sort of the unique characteristics of these sorts of data, and because there's systematic data collection, unlike, as I was saying, a lot of other big data problems. I personally find it much more inviting because the data are collected with hypotheses in mind, and one of the things that we can help do is really tie down those hypotheses, help sort of separate them out. But again, that requires the neuroscience insights as well as the statistical insights.
Yeah, I can't help interjecting one small addition to that, which you and I have talked about in the past, which is in the field, there's this tremendous excitement about sort of free behavior, or in the case of if it's a visual stimulus, it's natural scenes and unstructured. And now there's a big move to have unstructured stuff with mice or even with non-human primates. And what seems much more productive is to try to figure out a smart way to find the best features of these highly structured experiments and push them more toward more realistic experience, which is what people are really after. Because the one limitation of the high structured experiments that just people have been reacting to is they're sort of stylized. And so there's all this push, but I feel like they've thrown the baby out with the dishwater in a sense, and they've gone very far in the other direction sometimes. And it's like, I don't think they're going to learn very much from that personally anyway. So there's a huge challenge there of trying to think that problem through in a constructive way.
Right, but I guess the thing is, the structured experiments are not going to disappear tomorrow.
Right.
And there are tons of data sets which are, cataloged, which can either have been archived where, the thinking could be, the more structured analyses, in an experimental design sense could be exploited, completely. And yes, and I think, trying to, how does, as opposed to just showing showing in an animal like gratings in a certain orientation and changing them in some sort of artificial way, what does a natural scene look like? How does an animal, and that is the question to get to, no question. That's certainly the case. And as soon as you start mentioning feature selection, all the ideas of dimension reduction and dimension reduction in dynamical sense, come into play. So these are things that, statisticians are working on. And as you're pointing out, they come up quite naturally in a problem like this natural scenes question or natural stimuli, problem. So I think right there, again, there's a innumerable possibilities for statisticians to get involved in these sorts of problems and also I think, take the helm, make important methodology contributions, and I think make important, neuroscience contributions.
Mm-hmm. So, one thing that also has happened to me many times in like, I think the typical situations we're having dinner together or something, and maybe it's at a conference or some other place, could be over lunch. And we talk a lot about statistics and statistics training and so forth. And then the subject switches to anesthesiology. And I've watched you develop as both a statistician, and I've heard a lot about your development as an anesthesiologist. And it seems to me that for an awful lot of your early career, the two didn't have anything to do with each other. And then all of the hard work you did kind of in anesthesia, both as a clinician, as someone just kind of learning about it as best you could, and in statistics, likewise, working on a series of problems, but of course, neuroscience eventually. It seemed like this somehow it came together. And I don't know how long ago it was. It might have been as much as 10 years now. You started to identify these previously mysterious mechanisms that are involved in losing and regaining consciousness.
Mm-hmm.
So it always seemed to me, and I kind of like to hear you talk about these kind of dual interests and how they've interacted. Why don't we start with that? Could you just say something about that?
Yeah, sure. So you're right. So I decided that I wanted to be an anesthesiologist. My third year in medical school, I did my anesthesia. I did anesthesia for 2 weeks during my surgical rotation. I really enjoyed it. I liked the physiology, the pharmacology, the fact that we did procedures. I liked how anesthesiologists took care of patients. And again, a bit of serendipity. It was really, I decided late that I wanted to go into anesthesiology. I hadn't done a full month rotation. So Jack McPeak, who had actually, again, serendipity, had been on my undergraduate thesis committee, ran the medical school student rotation at Mass General Hospital. And so he arranged for me to do a month in July of, so that would have been July of 1986, to meet the requirements so that I could apply in anesthesiology. It was a fantastic experience. And so I knew I wanted to do anesthesiology. I knew I wanted to do statistics. I continued working on circadian rhythms once I, finished my training in anesthesiology. And so the two had nothing to do with each other. It was a day job and a night job. My research world was centered around circadian rhythms, starting to think about neuroscience data analysis. And, I took care of patients clinically. But I'll be frank with you. I kept this thing, which I call my BS list, because there was a lot of stuff in anesthesiology that made no sense. I mean, people would tell you all these things about, and they were based on these like totally sort of unfounded arguments, but they were the right answer to the questions on the board exam. So you learn. That's the way I viewed it. And the one thing that just bothered me was people would say, we knew the drugs bound to certain receptors. So to be very concrete, propofol binds to GABA receptors, or sevoflurane or isoflurane binds to GABA receptors. And then you'd say, well, okay, that's fine. We know what the molecular targets are, but why do you lose consciousness? Why do you have an altered state of, it doesn't tell you that, right? And so I just started, looking at the anatomy of the brain. But actually before I say this, I must admit I was pushed by one thing. I would go to neuroscience meetings and we'd talk about the statistical methods or analyses that I was doing. And then very often people would say to me, oh, but I hear you're an anesthesiologist also. I said, yeah. And they go, well, how does that work? And I go, oh, we don't know. And then, people would look at you and go, you mean you do this in the operating room, you're putting these drugs in people, you don't know what you're doing? and actually, after a while, after you hear yourself say that, particularly you hear yourself say that to neuroscientists who are really bent on understanding the mechanisms of how parts of the brain work or how the brain represents phenomena or information, what have you. I realized, one, that didn't sound too cool. And then the second thing was that the paradigms that, the Matt Wilsons that I was working with could be used to study anesthesia, because they put electrodes in rodents' brains. And then why didn't we just give anesthesia and just sort of see what the brain was doing? So so basically, as I hung out more with the systems neuroscience, I realized that the ideas that their approaches to studying problems could be used to study anesthesia. And so I came up with this idea of having not only rodent studies, but not human studies, non-human primate studies, doing them all at the same time to try to learn as much as we possibly could. And, this took a team effort. And, the good thing of, there were a lot of people who thought this was a fun thing to do. So I was able to, get a number of people to collaborate with me to, to pull this off. But it didn't link up the two fields, being an anesthesiologist, practicing anesthesiology, and, doing statistics research didn't link up. And then once we started, so I wrote out detailed discussions of how the drugs could act in the brain and the circuits to generate altered states of arousal. I did a couple of reviews, one in the New England Journal in 2010 and then one in the Annual Review of Neuroscience in 2011. And then around that same time, we began working with Nancy Coppell, trying to build models of the circuits and the dynamics that we were seeing.
Yeah, Nancy's a mathematician at BU.
Yeah, a mathematician and an expert in, dynamical systems. And I began working, years before with Patrick Purdon, starting to do human experiments to collect, data on humans and looking at the human brain under anesthesia. And the one thing that was just very apparent was that the drugs created oscillations in the brain. Every drug created an oscillation. The oscillations were basically mechanism dependent. So you had a different mechanism, or drugs in the same class showed similar oscillations. And as you change the drug class, you got, different oscillations. Mm-hmm. So this seemed to be like an entree into trying to get some insights into how the anesthetics were affecting the brain and how it produced these states like unconsciousness. And so what does that mean? You're talking about oscillations, so that puts you right back in the realm of like spectral analysis and time series analysis. So in that sense, I was, trained, to start looking at these problems. Once I realized there was this problem that was sitting right under my nose that I've been ignoring for all this time. And, and we've made a lot of progress, not by inventing like a lot of new methods, but I would just say applying a lot of basic methods correctly and diligently. And we've gained a lot of insight. So you can map out, you can categorize the anesthetic drugs, based on their drug class, because their EEG signatures or their local field potential signatures look different. They differ between class. They change systematically with drug dose, and they change systematically with patient age. And so right there, you have a framework for monitoring the brain under anesthesia, and also starting to understand how the drugs are acting in the brain to create those states. So that's how it eventually came together. I literally had a day job and a night job. And then when I was telling people in my night job what I was doing in my day job, and I realized what I was saying to them didn't make sense, I said, maybe I better fix this.
So the other thing that I wanted to comment on is it has always struck me with knowing you that there's a certain, I don't know, I just wanted you to react to this. I don't remember our ever talking about it, but there's a certain similarity in the knowledge base that you bring to bear as a statistician, I think, and maybe as an anesthesiologist, because at least for the kind of statistics that, I've been doing in neuroscience and that you've been doing, and I think it would apply in many other fields as well, you have to have a fairly broad background and a broad perspective. And that includes, obviously, statisticians have to know things that the lay person might think is computer science, right. Algorithms and so forth. We have to know something about, in the case of, in the case that you're talking about, signal processing, what's usually thought of as electrical engineering.
Right.
And we have to know something about how the scientific method works,
mm-hmm.
right? And then on the other hand, it seems to me like we always have this sort of broad view. We're also very conscious of interconnecting different fields, because statisticians do this all the time. You gave an example already of earthquakes and neural firing. That's just one of dozens and dozens of examples where you have a technique that's developed in one area and it transfers very nicely to a different area, completely different area,
right,
of science. And it seemed to me like when I heard you talk about anesthesia, that likewise, you sort of mentioned this already, you had to know something about physiology, you had to know, there's various pieces that you had to know that seemed to me like you might have had a real advantage in some ways stepping into neuroscience, because you had knowledge that that was broad. I mean, compared to many neuroscientists that I know, I feel like you have a far broader knowledge base, actually,
Well,
in certain respects.
Well, I don't know if I have a far broader knowledge, but there's something which there's a bit of expertise which I will say has been very helpful to me, and I've used, extensively, and that's physiology.
Yeah, maybe that's where I've seen it.
And so, in other words, you see these phenomena that are occurring, and they're physiologic principles that they have to respect. And so, in other words, I can't. It's hard for me to think about the drugs acting at circuits without thinking about the behavior that I see with patients in the operating room.
Mm-hmm.
I see, and so the reality is, I can do a neuro exam, and I can detect that certain parts of the brainstem, let's say, are shut off, and I can see the time course over which they shut off. So then that tells me something about where the drugs have to be acting.
Mm-hmm.
And so I would say that it's the physiology which is really, and that's what I'm grateful for having gone to medical school, because those principles we use over and over again in trying to, especially as we try to think through scales. Like we have this very gross behavior: someone loses consciousness, but then we dive down at the molecular level and say where the drugs are binding. But somehow we have to get from that molecular effect back up to the scale of the whole organism now,
Right,
being in a state of unconsciousness.
right.
So when we start new projects, like with my students, I ask them, I require that they go and review the physiology very, very diligently and come back and tell me a story. Tell me a story about the physiology of what's happening here. And then let's think about what we're going to do methods-wise or modeling-wise or that sort of thing.
Mm-hmm.
Because I see that in many cases, those constraints are overlooked. And then the other thing is, so then being an applied physiologist, having the experience of being in the operating room and just seeing these things, and not just theoretically, but then, having a kind of a sense of the time scale. I mean, one of the people that I think is a real master of this has been Warren Zapol. I mean, like Sam was mentioning, I'm the Warren Zapol professor. But Warren has this uncanny sense of just putting things into physiologic context when it comes to respiratory physiology. And then just, seeing how all of a sudden, a problem seems very complex to everyone else, looks very simple and straightforward to him. And so I can't say I've done these things nearly as sort of as elegantly and as deeply as he has, but it's that spirit, if you would, that I think has been driving it. So I think that's a small comparative advantage I may have had.
Yeah, I mean, I think that's probably what I've seen, and it just to me is very interesting, because the way I would phrase what you just said is, and I think picking up on one of the words you used is, you're automatically thinking about the whole organism,
right?
And that's very different than I mean, part of the reason is because science is so specialized, and neuroscientists, especially PhD neuroscientists who are not physicians, they're going to be looking at a very, very small piece of a big puzzle.
Right.
And the ability to step back and ask how this fits into the big questions seems important. It always is, right?
Very, very much so. And I think that if we're going to produce new therapies, new approaches to anesthesia, new approaches to monitoring the brain under anesthesia, we have to be able to extrapolate from these very, small scales up to the whole organism. And that link is physiology.
And it may be that I'm pushing an analogy that's not really very good, but I see in statistics, in terms of when I interact with experimental people, and this is true even before I studied neuroscience, I feel like one of the things we do, and that we teach our students to do, to the extent once they get comfortable, is to ask the most basic questions. Don't answer the question that the investigator thinks they want to answer. Push them about, why are they doing this? What is this about?
Mm-hmm.
How did the experiment get run? How are the data collected? All these sorts of much bigger issue sorts of things. Try to get the whole concept. And there's a way in which, successful applied people in statistics, they look for that big picture.
I think that's the case, and I think that's something that we do very, very successfully. But I think I would take it a step further. I think there's a whole nother level in which those questions can be asked and answered, and the field can be guided by, statisticians, if the statisticians become more vested in the field.
Right.
And enjoy being called a statistician, data scientist, enjoy that as much as being called a neuroscientist, and feeling that, wow, like I really care about this neuroscience question, and I want to put my mark on sorting out, how this particular brain region works, or how does this, this neuropsychiatric disorder come about, or how can we treat it, and I think that that's what I would like to see more of, and I think that it's the insights of putting together the physiologies we were talking about, like with the statistics, that has been behind at least the few things that I've been able to help sort out.
But
So this has been great.
This is interesting because now, for a second, suppose that you're a student in anesthesiology.
Mm-hmm.
The kind of experiments you were describing, Emery, it sounded like the principle of it sounded like a design of experiment type of a question. At the very least, you play with different parameters that go into the drug, and you basically observe the reaction by the brain.
Right.
Now, and you also mentioned that for a long time, you didn't know much about, or when people ask you questions about anesthesiology, you're like, I'm trying to learn the foundations of it, but then you contributed to the field through your statistics knowledge. And so that prompts me to ask, can now we take statistics to sciences and have students in sciences learn more statistics beyond the introductory, let's calculate the formula of the variance course?
Well, I think very, very much so. And like in this particular case, where the statistics played a key role was, so if you look at like this article we wrote for the New England Journal back in 2010 that I wrote with Ralph Leidig and Nico Schiff, we had these very detailed diagrams about the anatomy of the relevant arousal systems and the cortex and thalamus, and where the drugs were going to be targeting. And then, so that's just a neuroanatomical, neurophysiological constraints. They're just there. That anatomy is, or at least parts of it were old and cold, etched in stone in many respects. And then now you give the drugs. Okay, and so the thing that we then did was you give the drugs and you watch the response of, let's say, like the EEG. And you see these very, very rich dynamics. And I just have to comment on those dynamics. The EEG under anesthesia is the highest SNR EEG
signal to noise ratio.
Signal to noise ratio phenomenon there is. Because one, as soon as the drugs take effect, there's no movement artifact. You think of all the other things where the EEG is used. You have sleep, looking at patients in coma, behavioral studies, people use them now to do biofeedback and what have you. There's a lot of noise in there. The noise drops away. And roughly speaking, you have a phenomenon where the EEG moves from high frequency, low amplitude oscillations to very low frequency, large amplitude oscillations. And the oscillations are not subtle. It happens the same way every time. So whatever's there had to be a very obvious mechanism is the way I would say it. But the one thing that we did was we took time and characterized the dynamics of each of the oscillations for each of the drugs in great detail. So in other words, we just did the statistics, right, or we did it, in detail, so that we could then say to our modeling colleagues like Nancy Gopel, look, here for propofol, you have slow oscillations and alpha oscillations. The alpha oscillations concentrate in the front of the head. Seemingly, there must be some sort of, the anatomy must favor that. What could that be? What sort of model could generate this? So it was the data, it was doing the work with, laying the foundation, the framework with the sort of the neuroanatomy, neurophysiology, characterizing in detail the dynamics which we observed when we looked at patients or animals or what have you, and then saying, okay, your model is constrained because whatever you do, you've got to produce this. You've got to reproduce this. And then making predictions with those models and going back and doing experiments which, either verified or, refuted those predictions. So the statistics played this crucial role of, the constraints, of really establishing the quantitative constraints that really let us understand what the dynamics of the brain responses are, when these drugs are administered to patients or animals.
So this has been great, Emery. I want to switch gears now. I want to go talk about a different kind of thing, which is really some of your personal characteristics and your personal experiences. And I know Sam wants to follow up on this as well. So the path of every successful person results from some combination of talent and persistence and luck.
Mm-hmm.
So in your case, what struck me the most are two different, I guess, intertwined abilities. The first is your great patience and confidence in turning away from short-term gain and instead investing so much time and energy in learning what you need to know and creating the environment necessary to achieve the results you recognized as truly important. So could you comment on that first? And then there's the second one I want to get to.
Well, let's see. Patience. I guess when you decide to do both an MD and a PhD, I guess that implies a certain degree of patience just right there.
Yeah.
It's not
Huge
gonna,
amount of.
Yeah. But I guess,
But I mean, it's setting up the anesthesia. I mean, to me, what I watched happen, and I don't know whether you experienced it differently, was, there's a lot of uncertainty there when you start out. You don't know what's going to pay off. And it took quite a while before you got results. You invested a lot of time. And of course, you had a day job. You had 2 day jobs.
Right.
So it wasn't like you were going to starve. But nonetheless, you were putting a lot of time and energy into something where you didn't know exactly what was going to happen. And this is what I say, you had to be both patient and confident.
Right. But I guess what was happening is that I guess there were 2 things. I had this vision of having a team working on this. And I had this vision of, doing human experiments, doing non-human primate experiments, doing rodent experiments. And there were colleagues around who agreed to take on those various components. So it seemed feasible. That was one thing. And I think the other thing was there were a lot of people who were working on mechanisms of anesthesia, but nothing they told me changed in any way what I did in the operating room. It had no impact. Because as I was saying, just knowing the molecular targets or knowing the characteristics of the receptor and the various subunits didn't change anything. And so there was an incentive to try to do something which was going to really have impact. But the thing which was just so compelling to me, I mean, I realized, and then the first time we sort of said this was in this New England Journal paper that I referred to before from 2010, was anesthesiology was a subdiscipline in neuroscience. There was just a fundamental flaw in the way thinking in the field, was going. And I think it persists today. And I think it's one of the things that's impeded our making progress. We are a subdiscipline of clinical neuroscience, like neurology, neurosurgery, psychiatry, sleep medicine, psychology. And we have our impact by giving pharmacologic agents, but they're acting in the brain and central nervous system. And once I realized that, I was just like, this is the right way to think about this. I mean, and so despite all the other things that were going on, I just felt like this had to be successful on some level. I don't know what level yet, but I know I just have a feeling it's going to be successful. And in fact, when I applied for, I won a Pioneer Award, back in 2007. And my application was very simple. It just said, I'm going to use neuroscience to study anesthesia. So that was the kind of the breakthrough idea, if you would. And I guess that it was, and the other thing is, as I started doing little neuroscience things in the operating room, They just reinforced my intuition. Like, just for example, people in anesthesia are in a coma. It's a drug-induced coma. The EEG patterns look just like the EEG patterns you see in patients who are in coma because of structural brain lesions. And so I started using the neuro exam that neurologists did or do, you know, when they're evaluating patients in coma. I started doing it on my anesthesia patients. And the cool thing about it is that on a neurology patient, the patient has an injury, and it might be several days, several weeks before you see a change in the exam. Anesthesia, the feature is there, they put under anesthesia, it disappears, and you can see it come back. So it turns out it's the perfect place to teach the coma exam to like medical students, because you'll actually see the response or the reflex present, disappear, and then come back. So I'm sitting there watching these things and I'm saying, like, I don't care what everybody says, right? This is the way to think about what's going on here. And you know, it was just so it started to all kind of tie together. You know, the data analysis and statistics, the modeling that we were doing, like with Nancy, the clinical things that I was doing in the operating room, and then more recently, coming up with different strategies for dosing drugs, because as we monitor patients' brain under anesthesia, we can infer very reliably that they're unconscious at far less doses, far smaller doses of anesthesia than they're typically given. So I have to tell you this. So literally just last Tuesday, I was in the operating room. I took care of a gentleman who was quite sick. And he had to have a procedure it wasn't going to take very long. And the resident went up and got consent from his wife because he was quite sick and what have you. So we were very lucky. The surgeons were able to do the procedure in about 10 minutes or so. And we're ready to wake him up, you know, after about 10 minutes or 15 minutes under anesthesia. And he came to in about 2 minutes after we finished, literally like that. And he was very clear and what have you. And the resident said to me, he says, "Oh, I forgot to tell you. His wife said he always takes a long time to wake up from anesthesia." Right. And it's not that he takes a long time to wake up. It's just that he's overdosed. He's been overdosed before. I'm pretty sure of that, right?
Mm-hmm.
Right.
So then you're saying this confidence or what have you. I think about my circuit diagrams when I'm giving the anesthesia. I'm thinking about what this guy's circuits look like. He's, you know, an older gentleman, so his circuits are much more sensitive to the anesthetic. I cut the dose by about 2/3 of what you typically give someone. I can look at the EEG and see that he's totally unconscious. So it's starting to tie together the physiology, the statistics, the pharmacology. I mean, that's what happened. And so that's where, you know, if you want to, you know, call that confidence, it just started to tie together. So when I can think up an idea, you know, this theoretical idea about what the circuits are doing, and I can translate that into a different outcome for a patient, I think that's the part which makes me sort of more, if you want to call it confident, but more assured that we're thinking about this the right way. And there's a lot more to do. We haven't solved all the problems, but I think the paradigm of bringing neuroscience into anesthesia is the one that's going to really advance the field over the next several years.
Right.
But Emery, can you take us through your weekly schedule? I've asked you this question before, but I know that there are younger generation of readers of Chance or people who watch this interview. Like, what days are you actively a neuroscientist or a statistician, and what days you?
He's always a statistician.
I mean, I know that, but
Right.
I do only one of those, right? So I don't know more. So I'm curious about, and what sets of experiences, knowledge, skill you take from this job A to job B, and what are, you know, special to those jobs A and B.
Right. So what I do is, so my typical week, like for this semester is, so I'm teaching on Mondays and Wednesdays, teaching my undergraduate stat course. And then, you know, the balance of the day, I may have, you know, meetings. They could be group meetings or research meetings or or various committees. Most of them have recently been a lot of committees. Every Tuesday I'm in the operating room, and I try not to schedule anything like trips or anything like that for Tuesday because I don't like missing my operating room days. And then the balance of the week, like Wednesday, like Thursday and Friday, I'm, you know, again, meetings or either research or you know, or committee meetings, that sort of thing. So that's my typical day. So I'm 1 day a week in the operating room, and I've been doing that for like 20 years or so. And the one thing which I stopped taking call about 10 years, like overnight call. I stopped taking call about 10 years or so ago. And when I'm in the operating room, we try to make sure that I'm always working with the residents to do as much teaching as possible. So one-on-one, working, two residents, one in one, one in the other, going back and forth, helping them, supervising, their cases, for anesthesia, to try to, because so much of the learning that takes place is like, it's an apprenticeship, right? It's hands-on, like we're in a situation, how should we dose the drug? Like I told the resident right when we started, he said, should we put the propofol on 100? I said, no way, put it on 30. I mean, look at the guy's, look at his EEG. I mean, he's totally out, right? So that, that's, and then, so then the resident gets that feedback and actually sees and starts to have that same sort of intuition. So this is what, this is the experiential, sort of the apprenticeship type teaching that takes place in medical school, which is quite, or in residency training, which is different from, like classroom, classroom training. So that's that's my typical schedule.
So the other personal sort of characteristic that I've commented on, and many times, like in these settings where I've introduced you and stuff, is your ability to keep moving towards your goals, which is required not only foresight and persistence, but also this other thing, which is a kind of dexterity in sidestepping the inevitable pitfalls. There's pitfalls really with every project, and you've been really unusually good at sidestepping them, in my observation. And it's been many, many different kinds of pitfalls. Mm-hmm. So you emphasized the importance of this years ago. When we invited you to give an informal talk at Carnegie Mellon to a group of underrepresented minority students.
Right.
So could you describe some of the specific challenges you faced, the kind of thing you told them, in your personal life early or maybe subsequently, that would relate to the kinds of things that they would have been concerned about, and then the importance that you've attached to dealing with those situations productively?
Yeah, I mean, I think that, I guess one of the things that my mother used to tell me is that nothing is supposed to work. You have to make it work. And, that requires, putting in, time and energy. And I just remember, I think I'm quoting him correctly. I remember Brad Efron saying one time, you really don't know how smart you are until you work hard. Right. And I really like think that's the case. So for me, what happens is that I get interested in something like statistics, and I can't imagine myself doing anything else. When I decided I want to be a statistician, I thought that was the coolest thing that I could be. And I can tell you now, if I were about to start over, I would do exactly the same thing. I'd go after statistics. And so I guess what I'm saying is I develop a passion for it. And once I have the passion, nothing is basically going to stand in my way. Nothing is going to keep me from essentially achieving that goal. I wanted to be an anesthesiologist, basically the same thing. I wanted to go to, I wanted to go to medical school. And I think that, so there are going to be people, for various reasons, whether it's race or just because they don't like you, who are going to perhaps oppose what you want to do. So that's a given. The world is just set out that way. But, you can't let them stop you, because if you're thinking about solving important problems and you're going to, hopefully, make things better for, humankind, because we improve healthcare and through improving anesthesia, then, you have a bigger goal that you have to, you have to address. And, a lot of times what people want to do is to try to take you off the mark by engaging you and getting you into tussles and all these sorts of things and arguments and what have you. You don't have time for that. It's just like, okay, that's fine. That's what they want to do. I want to figure out how anesthesia works. I'm going to keep working, very, very hard on this. Or I want to get my PhD in statistics. So I'm going to keep working very, very hard from that. And the other thing which I can also say is that I was, very, 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 very fortunate in like working with somebody like Professor Mosteller early on. And I think in working with him, he came away with the impression that I was very serious and very focused and wanted to have this happen, wanted to become a statistician. So, I think that knowing that somebody like him endorsed me and felt that, that I was, being successful, even with this very small problem that I worked on in my undergraduate thesis. And then again, the same sort of thing. I said, Jack McPeak, who was on my thesis committee, he helped me get to do the medical school rotation so that I could, become an anesthesiologist. The point being is, if you're doing a good job, eventually cream does rise. I really believe that. And it's not gonna always happen like right up. But the thing is, you can't let, the naysayers sort of dissuade you from pursuing your goal. You just can't do that. Because, if you're on this track and you have a goal that you're going after, like in my case, again, statistics, anesthesiology, neuroscience, just you have to stick to it. Because and the thing is, in a lot of these cases, particularly like for the statistics and neuroscience, or for like the neuroscience and anesthesiology, it was very clear that those approaches were going to have impact on the field because they were lacking. Good statistical approaches were lacking for neuroscience. Neuroscience was lacking for anesthesiology. And so that insight and the confidence to go through with it, I think, are the things that have, like I was talking about before, have spurred me on. So I guess in essence, it sort of boils down to, you have to have confidence in your abilities and confidence in what it is you're pursuing. And that comes with time. And again, I can, and we know that, it's just like a lot of, many underrepresented minorities, African Americans in particular, because then obviously that's my community that I know quite well, are often discouraged. And it should be just the opposite. We have to encourage, we have to build that self-confidence the same way that that I see in other students. And I think that's, for me, something which I've been, privy to have happen, because I think it goes back to, my parents, for example, always it actually goes beyond just my parents. I was telling my wife this the other day. My parents they had certain expectations of me and my brothers, expecting us to be successful. And then that translated to people in the community around us. Like I remember at my church, people saying Brown, they always call me by my last name, Brown, I know you're going to do something big one day because, you're always, you're working hard and you're smart, that sort of thing. Or like, Brown, I know you can't go off and do anything bad because, your parents will be all over you, So their expectations, so these expectations start to reinforce themselves. Like, I'm supposed to do something big. I don't know what it is, but I'm supposed to do something big, like you walk around with, like with that idea. And, and it starts to, and again, I can honestly say that there are far more people who I can think back of who've been extremely helpful to me than the people I can think back who are like impediments. Because, and actually one of the people who's actually been most helpful to me throughout my career has been Rob. I mean, at multiple junctures. Like, for example, the first major presentation I gave was at Bayesian Statistics Symposium that Rob organized back in the early 90s. And I talked about, measuring blood flow, through the human body. And then, so I believe cream rises. I mean, it sounds kind of trite, but you can't underestimate how hard you have to work. And I think what Brad Efron said is correct. You can have all the talents in the world you want, but you still have to work hard to make things happen. And I guess I've never been afraid to put in the extra time, to sort of make things happen. And I don't see any substitute for that, because nothing is just going to happen magically.
Okay, so Emery, I just want to push you a little more on this, if you don't mind.
Sure.
So I was saying that my wife and I, Loretta, who we were talking, I think because I was thinking about doing this interview with Sam of you a couple nights ago, maybe, and she immediately said, "Oh, I remember that time that he talked about, his personal experiences at that dinner."
Mm-hmm.
And interestingly, so I think you might have told two different anecdotes, because I feel like she immediately brought up one that was slightly different than what I had remembered. But just to get the record straight, so you started in segregated schools up until age 9 or something, like 9 or 10, something like that?
I was in segregated school up to 6th grade, yeah. Okay. Yeah.
And then you told some story that highlighted, again, a kind of impediment or something that made you, something negative was directed at your way, toward you. And you were talking about basically how you were able to dodge it in your life at that early age.
Right.
And then she had another thing she was remembering. I think you told something about there was a prize that had to do with languages, and that you had to shift from one language to another. Maybe your mother intervened or something. I don't remember the details of that either.
Yeah,
but I think there were two. Those are the two that might be nice to review, just to be concrete.
Well, I can tell you about the language. The language prize was. So what happened is that so there was a summer program that my city had with they had a sister city in Colombia, which was Sincelejo. And so every summer, students from Sincelejo would come to Ocala, and students from Ocala would go to Sincelejo. It was a big deal. And it was the students who were who were in second year Spanish,
in high school.
Second and third year Spanish in junior high.
Oh, junior, okay.
Right. And so, during my 7th grade, I went to school for the first time with white kids. I'd never gone to school with white kids before that. And I went to school in Ocala. It was freedom of choice. That was the way they integrated the schools. And what freedom of choice meant there was a few black students who elected to go to the white schools, but no white students elected to go to the black schools. So I went to school in town, a junior high school, Osceola Junior High School. I did pretty well, grade wise and everything. It didn't have any problems. Didn't change what I was doing. And the students realized that I was quite capable. In particular, I was quite capable in language, because I really wanted to study languages, and that was the first time I had a chance to study Spanish. And so then the next year, I could apply to be part of one of the students who would go to Sincelejo, and I hadn't been out of the country at that time, and this was a big deal. I was always thinking about that. I kind of had that on my list of things to do. And so what happened is that I wrote my application. And in my application, I actually said, I had this thing for words at the time. I was a little bit sort of, I like to use a thesaurus, let me put it that way. And I said something like, it would be an edifying experience for me as well as my Colombian counterparts. I remember that expression because, so I was at a football game, a few weeks after I'd submitted my application. And I remember Sidney Morthland, who was one of the kids I'd gone to school with. So I was back at my segregated school then. And I was at this football game, and I ran to some of the kids I was going to school with the year before. And Morthland said to me, he goes, yes, he said, Mrs. Peterson read us your essay, and he told us about how you would find an edifying experience for you and your Colombian counterparts, right? Like, right. And everybody knew who, like, the top student was as far as languages was concerned, because this was something I was really into. And so my mother went to a meeting. She was part of the League of Women Voters, and they were discussing the whole policy to send students down to Sincelejo. And she said, I don't want you to be disappointed, but they're not going to pick you. And they've decided to draw a line saying that, you know, only students who are actually going to school in Ocala and not in the county are going to go to Sincelejo. And, you know, I was,
in other words, white students.
Right.
yes. And so I was like, I mean, I was bummed. I mean, I was totally bummed because this was a big thing. But then what happens is, you know, the next summer, I took French because they didn't have at the school I was going. So now I was going to an integrated school in the northern part of the county. They integrated the whole northern part of the county. So now I was going to integrated school. I was in 9th grade, and they didn't have a Spanish teacher, so I had to stop Spanish. So I started taking French, and I was into French. Wow, this was so cool. This was, you know, and so my French teacher says, you know, there's a summer program, you know, for students who want to go to France. You know, you have to pay for it, but you know, if you're like so into this, and you know, maybe you should do that. And so I brought it home to my parents, and I said, oh yeah, so we'll send you to France. So I was thinking, like, you know, so I didn't go to Colombia, you know, but now I've gone to France. And then the next summer, you know, I went to, you know, Phillips Exeter Academy, like the summer school. But the point is that they weren't going to stop me, you know, just because I couldn't go to Colombia. And in fact, it's rather ironic, because now I've been to Colombia twice, you know. Now I just went I was there just last year. And the thing that I do, like when I go to Spanish speaking countries, I give my lectures in Spanish. I mean, so I finally made it to Colombia, you know, made it there sort of in grand style, in a sense, you know. And I think that, you know, the one thing, but you know, when that happened, you know, I mean, I know I was quite disappointed, but I still knew I was the best student. They couldn't take that away, and they knew it too, right? And I've had to remind myself of that on several occasions, right? It may not show up now, but they know it. They know it deep down, you know, and that doesn't change. That the facts of the case don't change. And so, and I can honestly say I've had to rely on that sort of internal conviction, you know, saying like, you know, you've won the game, even though everybody else hasn't acknowledged it. And that's been very, very important for sort of keeping my eye on the prize in many situations, because otherwise a lot of people would be, you know, just sort of flustered and, you know, oh my God, you know, like unfair, and like, you know, ranting and raving, you know. And sure, I think I had a, you know, like a strong case, you know, to maybe argue if you want to have the best students, you know, representing Ocala. This, I was certainly, let's just say I wasn't the worst. Let me put it that way. You know. So but I mean, those sorts of things play themselves out over and over again. And the way they play themselves out now, they don't play themselves, well, I mean, more recently they have played themselves out very overtly because of the certain social climate we're in now. But, you know, more commonly, particularly in professional settings, they play themselves out in microaggressions. Mm-hmm. And, you know, knowing how to deal with, you know, microaggressions, because it's very easy to, it's very, very easy to use those things to take you off your game. And they can be very simple things. Like someone comes into the room, there are 3 people in the room and you're one of the 3 people. They say good morning to the other 2 and don't say good morning to you. Now, they can't say they were racist, right? You know, but again, you see what the dynamic is like. Fine. I was having a good morning before you came. So, you know, it doesn't bother me. But, and I think that, you know, doing some planning, you know, for situations, sort of thinking through some of these situations preemptively is very, very important, because there's certain battles you do have to win in order to be successful. So I think that, I think that having the confidence in yourself, it comes back to that. It really does. Whether it's with the languages, whether it's with the Spanish, with the languages, whether it's with the neuroscience, whether it's with the statistics. I think that's the part which, and I think I know, in fact, that stems from my parents, because they were always encouraging me, just hang in there, just you're doing, more than well. Or being frank with me, like when they told me that it'd be a good idea for me to go to Exeter, Phillips Exeter Academy, because to leave, the high school there in Florida, because I really wasn't being tested. You're being successful, but don't fool yourself. There's a whole nother level up there which you should be playing at, and you haven't demonstrated you could play at that level yet. So you need to think about that. So that's I think that might have been I remember telling that story about when I was visiting you at Carnegie Mellon, and back when we were speaking with the group of underrepresented minority students.
There's one thing I feel like it's reminding me of, and I'm a little embarrassed about this, but I'll say it anyway. And I don't know whether you remember this, but there were, and I'm hesitant to even say what job this was. Let's just say you had taken a new position,
mm-hmm.
and you said to me, because we were friends, you were talking about your colleagues. It was not long after you had kind of started to settle in there, and you said, they don't really know what to make of me. And I looked at you and I said, you mean because you're so interdisciplinary? And I don't know what you said, and I don't remember that part, but Loretta, my wife, she's like, she used to call me naive. Now she calls me privileged. when that kind of stuff happens. But, again, it was like something you had to deal with, presumably.
Yeah, well, I know exactly what you mean. What I was referring to was the fact that, let's just say African Americans in academics, particularly African Americans in the sciences and academia, are not a dime a dozen.
Mm-hmm.
And a lot of for a lot of white people, the African Americans that they know of that are successful are people in entertainment, in the sports, or they're, or they may be very successful, but they're on staff at the university. They're not the professors of this sort of thing. And so yeah, it's like, they don't know whether, should they high five me or say, say, hey man, what's going on? Have like a normal conversation, do you play golf or, something like that. So yeah, I mean, that's like very, very real. It's like, whereas, I've been around, hundreds of, white people in those situations, whereas in the other, looking at it the other way around, for them, this is a rare encounter. And that's just part of the reality. And so, but you can often, you can often see it. And it shows up, sometimes where, like, even, I remember one of the first times I was going up the elevator, like in the neuroscience department at MIT, and this guy says to me, like, whose lab are you in?
Did you say mine?
I did. I said my own. He goes, oh. I said, that's my lab, right? Everything. Oh, I see. Oh, and he, introduced himself and told me what he was doing, and he made it a point to tell me what he was doing, and that he was working not only in Boston but also abroad.
Yeah, so, just to get the pecking order right.
Yeah, so that's great. That's fantastic. And then what was so funny was about a month or two later, he goes, Emery, there's a job opening here on the faculty. I was wondering if you could put in a good word for me. I go, oh yeah, definitely. I remember you. Yeah, definitely.
I thought you were going to say he's knocking on your door because he wants statistics help.
Yeah, no, he wanted the job, or like I was coming upstairs once. I finished teaching and I was walking up the stairs and I kind of had my folders, which were sort of falling out of my hand. I need to get my wallet out so I could open the door to get back into my area there. And this guy comes up to me rather belligerently and he goes, "Where are you going?" I said, "Just watch me, just watch me." So but it was very clear what the intent was. Like, what are you doing here? And dude, I mean, so, or, another example, like I came to the operating room one day and they said, "Emery, the first case has been changed. The first case is a guy who now weighs 400 pounds. And, he has some sort of abdominal surgery." Like, oh my, he said, the resident is up seeing him. So I said, "Oh, I've got to take a look at this because 400 pounds is 400 pounds. So I went up to see him and I come upstairs and the resident is there seeing him. And I said, "Are you happy? Anything I should know about it?" So he gives me a little report. And so the nurse comes out rather gleefully and she goes, "Oh, are you here from transport?"
Oh, God.
And I said, "No." And so the resident goes, "That's Dr. Brown. You should Google him." Right. But I mean, those things, I mean, and this wasn't, like that long ago. This wasn't like, 20 years ago or something, So these, again, there's this reality out there, which I can't ignore. And how you figure out, but the thing is, you can't let it take you out of your game. Because, getting in some sort of ridiculous argument with her, and I mean, that's exactly the sort of thing you don't need, right? But then, having the resident come up and say, "Well, look, when you have a second, pull out your cell phone and, just sort of like see who you're talking to there." I mean,
did she ever apologize?
Oh, no. She was happy about her. She's like, "Oh, I thought you were from transport."
Oh, wow.
Yeah, I was like, okay. Yeah.
Clueless.
Yeah, or exactly. Yeah. So
I guess what you're saying is your trick here is just don't let it change your mindset.
You can't let it change your mind. Can't let it get under your skin.
So as we were talking about this, do you have any suggestions, Emery? Any practical solutions? We're doing this for American Statistical Association.
Mm-hmm.
There are committees being formed on, promoting statistics for underrepresented individuals and so forth. But what practical ideas, solutions that come to mind to promote statistics and sciences, in broadly speaking,
right,
to minorities?
Well, I think that I think the obvious things. I think and just doing the obvious things and following through on them. So first of all, while it's great to have, it would be great to have more, African American mentors, or maybe mentors of who are Latinx descent or this sort of thing. Those don't have to be the mentors for these things to be successful. For example, the program to really integrate Harvard Medical School was actually formulated and driven by essentially 4 white guys. I think a large part of it is just having the heads and the hearts in the right place and sort of doing the right thing. Now, clearly having role models and examples, it's like, I forget the woman's name, but she just became the manager of, I think it's the Marlin. She said, if you can't see it, you can't be it, you know, sort of thing. And that's true to a large extent. So I think that, you know, realizing that, you know, so even people who are not part of underrepresented minority groups can be mentors and very valuable mentors. And arguably, there are more efforts which can be, in other words, there are more possibilities there than there are for having mentors who are underrepresented minorities, because they're just fewer. If we have to wait till they're underrepresented minority mentors, it's never going to happen. It's never going to change. All right. So I think that's, so in other words, so that means the responsibility is part of society's responsibilities and not just, you know, on the underrepresented group itself, you know, to sort of bring about these, to bring about these changes. And I think that, you know, the pipeline issues are very real, but they can be fixed. You know, the summer programs work. You know, the post-bac programs, you know, work to get, you know, students into statistics. And, you know, you know, the one thing that, you know, very often, you know, at least in the African American community, people think about going into medicine because they really want to help people. And I know, like in my community, you know, one of the most respected, when I was growing up, one of the most respected people in the community was the doctor. You know, there were like two African American doctors in my community. Mm-hmm. Well, you can do important things and have them impact the same way as a statistician, you know? Because I mean, think of the drug trials, which are just finished, right? Somebody has to sign off and say that this was actually properly done, the inferences are correct, and that's not that's not the infectious disease specialist, you know, that's the statistician. Right. And so in other words, so realizing that these jobs can be just as have an impactful on society as, you know, physicians, I think is something which we just need to, you know, emphasize. And I think that there's no substitute for doing the hard work. There's nothing magical. I mean, we have to create the environment that, you know, that makes it possible for, you know, underrepresented minorities to see just like anybody else. And I think that's totally doable. And when you look at, and, you know, there's some interesting case studies where this has been made clear. You know, the University of Maryland, Baltimore County has had an amazing success rate at getting African Americans into the sciences and the mathematics. And actually, I've had several of those, at least two of those students in, you know, in graduate school, like at MIT there. And there's, you know, there's nothing about the soil in Baltimore County that's any different from the soil anywhere else, right? It's just that Freeman Hrabowski has just decided, I'm going to make these guys successful. Mm-hmm. And so they come in with this attitude, like, we can do this, and they're part of a community, and they've been successful. And so I think that those sorts of efforts can be reproduced and propagated, and they will yield, you know, they'll yield fruit in the very
near. That example from Baltimore also requires money, structure, and management.
Yeah, but he's the president of the university.
So grants, supports.
Yeah, but this is all stuff that people want to do now, right?
Right. Exactly. So, for example, I'm on the board of the Simons Foundation now, and Jim and Marilyn have said they set up a program where that anybody who has a Simons Foundation grant, anybody, can get a supplement to take on an underrepresented minority postdoc or graduate student. Mm-hmm. The same way you have minority supplements for NIH grants.
Got it. Yep.
So you know, the resources are being created.
Right.
And I think that, you know, and I think it's just a matter of putting in the effort to help direct the talented people in that, you know, towards those resources. So I mean, I see that. So I don't see anything magical. I just see I just see, you know, really just following through on this list of things that I think a lot of people are talking about now, and then just committing to them, and then seeing where we are in 3 or 4 years or 5 years. I think we'll make a lot of progress.
No, this has been a great conversation, Emery. This is, I think this is going to make a really great interview.
Let me thank you, Emery, as well for participating in this. This was a fantastic interview.
Oh, you guys, thank you all.